Are you ready to go?
Yeah.
Would you like to stop screen sharing so that we can see Gary? Is that possible?
Yes. Thank you.
Hi. So this is a soapbox about a topic that emerged from the data modeling and integration breakout session in the network modeling subgroup. And the topic is expanding cell-cell interaction models to include long-range signaling. So we discussed how networks have traditionally been used to understand how the cell works, focusing mostly on intracellular networks. And more recently, single-cell genomics has opened that up and enabled mapping of cell-cell interaction networks that are already quite popular. And we wondered how far these networks can take us and discussed their use in understanding tissue-level structure and function, like what's the function of a gradient, which is made up of a group of cells, all the way up to physiological systems such as endocrine systems, explaining circadian rhythms and the gut-brain axis. So we realized that many of these systems have been studied, for example, in the area of computational physiology, but single-cell genomics may bring a molecular mechanism perspective to these processes, and we don't really have any computational physiologists in our community, and we haven't linked to them. So maybe we need to link to that community. We also discussed ways that we might need new languages to describe functions of groups of cells, or generally how we can integrate multi-scale thinking, systems biology thinking across the Human Cell Atlas project. So we'd like to, the next step for this topic is to discuss how to connect with these other communities. Maybe we need a workshop with computational physiologists. We'd love to know if anyone's a computational physiologist who's at this meeting, and also if anyone has links to computational physiology community, please, I guess, text me, contact me if that's the case. We created a new Slack channel on the HCA Slack workspace called Cell Ecosystems, Cell-Ecosystems. And we want to just continue discussion there. So please join that Slack channel on the HCA Slack workspace if you're interested in thinking about cell ecosystems all the way up to physiology. Thank you.
Yeah, great.
All right, go ahead.
All right, so we're reporting from topic group 15, which was our standardizing biospecimens. First of all, we just reconfirmed our commitment and belief in the benefit of open sharing of ideas, samples, and team science. And my co-moderator is Todd Valerius. So I'm going to talk about our first two. We had 4 topics. Our first two. The first, we talked about just sharing biospecimens, and we very quickly got into identifying the barrier, which is overcoming the legal and institutional barriers to sharing tissues and potentially identifying data. So as we started to talk, we found that these barriers seem to concern issues like the content of consent documents and process, IRB and legal department interpretations of federal regulations, institutional and investigator concerns for financial, academic, scientific return on their investment and willingness to share. And finally, understanding what the barriers or lack, we're not really very well informed about the barriers in sharing materials between countries. We recognize the benefit for crosstalk across the consortia and programs to share best practices. So we'd like to take advantage of this consortium that has been meeting these 2 days to work out the challenges in the regulatory concerns around tissue, biobanking, and sharing of tissues. Ideas such as a common consent form that can then be shared with regulatory agencies like NIH, who might adopt them and help our institutions grapple with those stumbling blocks that they feel they can't otherwise meet. So each consortia has been grappling with these questions. I just came from the Ethics and Diversity Working Group, and a very similar concern was raised. So we would like to suggest that we develop a cross-consortia working group to make collective and global progress. The second point was deciding on and/or creating a central dynamic protocol repository. So in that discussion, we acknowledged the benefit of a unified, a place for unified vetted protocols and opportunities to share evaluations and QC on those protocols. We suggested that sharing protocols in a central manner in a place where dynamic feedback can be given would be best. We suggested that the cross-consortia decision-making take place that would decide where that protocol repository exists. Protocols.io was brought up a number of times. Okay, to give Todd 15 seconds, we are looking for volunteers and we need a way from KI to a mechanism to volunteer for those two working groups. Sorry, Todd.
That's okay. I'd like to add one thing from my side of the discussion.
Yep.
We talked about establishment of a virtual cross-consortium biobank. We mean virtual because it's increasingly clear that there are useful biobank samples across the various consortiums. And we think if there's a way to discover what biosamples may be available to other consortium, there could be sharing of those. So the goal was to, again, a cross-consortium virtual biobank, a VBB if you would, which would push forth a meta standard that we would get from already operational meta standard working groups that are across consortia, and then put that to work and give people the ability to find additional biospecimens that are going unused at the moment. And then from that, hopefully we will establish perhaps a benchmarking set of samples that would be used from, and again, this will be in our blue box document, a large number of tissues from a single specimen, like it's been done in GTEx, cadaveric samples, for example, that many people could come and access, an open, accessible, free of too many of the structures that Gloria alluded to, so that people could standardize around a sample to validate their technologies. And our ask is that we create some funding mechanisms to move forward. Yep, that's what I got. Thank you.
Okay, lovely. It sounds like, Jonathan, go ahead.
Is it fair to say that I've been hearing a clicker in the background? Is that you?
I didn't hear a clicker. I don't think it's me. Anyway, it wasn't a timer. That's for
we
sure.
got somebody
Thanks
typing.
again. Yeah, okay. Someone might be typing. If you are, please mute. Thank you, Todd and Gloria. It sounded like you covered two in there, if I'm not mistaken. And so please do set your timer. That would be useful if you could self-track, because I hate interrupting you. So if you could do it now. We have Amy Bernard coming up next, or Bernard, however she pronounces it. Either one, lovely.
Either one is fine.
Christina Curtis is on deck, Christina. So Amy, please take.
Okay, so let's see. I'm summarizing for session 12B, which was defining data types for each taxonomy level. And I'm not so much summarizing as highlighting a couple things that kind of bubbled to the surface that we're excited about. One of which was that there's a lot going on already, and sort of doubling down on the communication streams between what kind of solutions are being used across the different organ systems and organ types to promote better understanding of what those data acquisition modalities are is ever important. So some of those were highlighted. They include particularly imaging modalities. So everybody's using single cell characterization at the transcriptomic level, but starting to really interleave in the imaging modalities that can start linking these together. We're using combinatorial strategies to link together, say, a transcriptomic label that can then be acquired or marked with some sort of imaging strategy, and then using these to map across different scales. So recognizing that it's a challenge, it's also an incredibly important challenge, and in order for the different modalities to be linked together, this is essential. So in terms of taxonomies, ontologies, and annotation of this data, everybody as well recognize that within organ systems, there's a lot of work already underway, and there's a need to sort of bring this up to another level so that the different organ systems are working with and refining and iterating on the ontology and nomenclature that is appropriate to cross-link across both these different scales and across an entire body system. And ideally also across time and across species when possible. So let's see, then there was also passion about the idea that some sort of meeting or summit of imaging people should get together to talk a little bit about the practicality. This was spearheaded by Alain Chiratal in terms of getting together people to talk about where compression or best practices for image annotation can be moved forward. So those were key things. In terms of governing strategies, where centralized, it's best. And so we call out to any sort of funding agency that wants to work with the team to come up with a centralized strategy that can organize some of this information. I think you might hear this through other breakouts as well, that a desire to centralize around nomenclature, taxonomy, and ontologies at a sort of centralized governing body would be ideal.
Very lovely. Thank you very much. Christina Curtis is next, and Katty Borner is on deck. Christina, are you with us?
Yes, great.
Floor
So
is
I'm
yours.
can you hear me? Summarizing for 12C, which was also on Atlas integration. And I think some of those themes cut across here. We were quite enthusiastic as a group about thinking about cross-Atlas cell state curation to really augment some of the cell type annotations that are already underway, and dually to mapping that into tissue ecosystems. So really the key here is that these cell states are dynamic, that we need to move beyond ontologies, but obviously use some of that framework, and then to map this really onto the tissue architecture. So I think some themes that we just heard really cut across this as well. Some of the take-homes for this were to begin to develop working groups that engage the anatomic pathologists with those that are doing these kinds of imaging approaches and modelers. And some of that's happening within individual atlases, but to cut across really is going to be necessary. So we're interested in working with others that are excited about that as well. And really we view the sort of long-term goal of this is that if we're going to integrate such data, we need to have a standardization, a uniform framework, and of course one can begin to then integrate these multimodal measurements. So we'll try and put forth a call for a working group around this and synergize so it's not redundant with other efforts.
Thank you. Finished? Lovely. Okay, and next we have Katty Borner, and on deck, so he can get ready, is David Osumi Sutherland. Hi, Katty. Go.
Hi, good afternoon, everybody. So this is for 6 CCCF user interfaces and anatomical structures and cell types session. This is with Peter Hunter from Spark and Jim G, BICCN. We are interested to see if we can agree on anatomical structures and cell types tables for 5 to 10 organs. There's an initial table that was originally published by KPMP in a paper which is entitled A Multimodal and Integrated Approach to Interrogate Human Kidney Biopsies with Rigor and Reproducibility. I have put that also in the group chat in case you have not seen such a table before. And it's table 5 in that paper. And that table is incredibly valuable because it shows the gross and subanatomical structures, but also the cell types inside of those anatomical structures. And it is even have marker with genes. And you could add additional columns to that table to also account for antibodies and stains and other assays that can be used to identify cell types. So if we could agree for a few organs on some of those columns, that would be wonderful and very valuable for the CCF effort. In our session, we started to create a larger table of different organs and different consortia working on those organs. And there's a tiny URL in the chat text, which you might find useful to just see if your consortia is correctly represented. So my call for you would be to go find that tiny URL, check for your consortium, and make sure that all the organs you deeply care about are checked off on there. And then if you're interested to learn more about this effort or be part of it, we had great discussions, and it's not just the 3 of us, we had a full house, so to say. We would love to welcome you. There will be a debrief on or around April 9. And to get an invitation, please just go to the CCF session and leave your name at the end of the document. And again, that link is also in the chat text. Thank you all.
Thank you very much, Katty. We have David Osumi Sutherland next and Dennis Shapiro on deck. David, are you with us?
Yes, hi.
Yes. Go.
You're muted.
Oh, you're muted, David. We heard you for a second. There you go. Okay.
Sorry, I don't know how that happened. Okay, reporting back from 12A on multiscale ontologies. I think there's probably quite a lot of overlap between this proposal and some of the other things discussed, so we might want to think about merging. The basic take home was that we wanted to have a series of organ-specific jamborees or potentially tissue type specific jamborees that go across organs, but I think organ specific first of all, with two aims. One is to improve and extend existing multiscale anatomy ontologies, and I think probably Ubron is the obvious candidate there. That would involve a certain amount of training work to get the community to be able to easily contribute and give feedback and review and improve what's there, and discuss how we might extend to add more spatial information. We'd like to do that in coordination with the CCF, with the common coordinate frameworks. We have ways to make these ontologies much more accessible now by making slices and by generating simple tables for review. So I think there's, the time is right for that. But then the second was to discuss how we develop data-driven methods for extending these ontologies, and that was a little more ambitious. There was discussion of how we might incorporate spatial information, cell type interactions and arrangements within tissues into that. So that's basically it. It's a series of meetings to try to review what's there and improve it and work out how to extend it in data-driven ways. I should say this is very similar to what came out of the cell type meeting yesterday. So those things should probably be coordinated if there's a series of meetings there. Again, need for review by the community. Okay, so I'm done. Thank you.
Thank you, David. Dennis is next, and on deck we have Nick Nystrom and Jason Swedlow. Dennis, are you with us?
Yes. So thank you for the opportunity to moderate the session focusing on antibody-based imaging platforms and how to analyze this type of data. And we had lively discussions about all the different platforms and also the different algorithms that are available. And there's no kind of ground truth or no way to compare different methods that we have available between the different technologies, between the different tissue types, between the different antibody panels. So what we were suggesting was to create kind of a challenge to find cell types and also identify unique neighborhoods across all the platforms. And we have a really good time to do this because HubMap and H10 both have initiatives going on where we get a lot of data from different centers actually collecting this type of data. So the next 3 steps that we came up with is, on the one hand, to create a common database for data storage and visualization. So that would also allow us to kind of standardize the formats and also enable really kind of a stress test of like, how does it look like once you upload the data? How do you visualize the different types of data? What metadata is necessary? The second part is really to look what technologies are available, what antibody panels overlap, and what antibody panels don't overlap, as an example, and then which tissue types are available. And then the third one is to create kind of a sandbox. And here we had some suggestions that Emma Lundberg showed us. I enjoy IO as an example, as a sandbox in bioimaging IO. And that's also in the Google Docs if you want to check it out later. So that would provide a sandbox for the different teams to just play around with the data, try different algorithms on potentially their own data, but also on other types of data. And the fourth one is really to create a ground truth annotated based on H&E, IHC, or the multiplex imaging data sets, and then use this to create a challenge for not domain experts so that we could give it to the image processing community and they could come up with novel ways to look into the data.
10 seconds. Yep, you good? Perfect. Thank you very much, Dennis. And Nick and Jason are next, and we have Mary Ann Martone on deck. Nick and Jason, are you with us?
I am.
Yep, here too.
Go to the screen. I don't see Jason, but we were in the session on how to analyze distributed data sets. And as you know, HubMap itself is a hybrid infrastructure. So on-prem and cloud, so we've been thinking a lot about this. One of the key takeaways we had was we've been waiting for this meeting to look into ways that HubMap and HCA might work together. There was a call recently from the NIH Common Fund Data Ecosystem. It's NIH-HCA 20005. And we've been thinking about ways that HubMap would work with other consortia within NIH, specifically Spark, Kids First, possibly UDN, and others. But we'd also like to look at ways we would operate with HCA. One of the keys here is really look at ways to bring our data sets together. Not so much in, never start my timer, never so much in developing a standard for data formats. That's not going to happen. It's too heavy a lift. But looking specifically at individual data types we might want to bring together from different efforts and to address specific medical questions. And so I think one of the follow-ups from this meeting would be to facilitate, and I'm looking virtually at no innovation for this, to facilitate an ongoing discussion with HubMap and HCA about ways that we would bring certain kinds of our data together, whether it's single-cell genomic or otherwise, to look at specific medical issues. That could be heart, lung, kidney, liver, whatever. And do it in a pilot sense, things that we can knock off in 6 months or a year to demonstrate success and learn from it.
30 seconds.
A related issue to that was, well, there are two things. One is not looking at a standard for data, but looking at tools for data interchange so that we can work with our data in whatever it happens to be. The second idea that Kati actually suggested was really good was now that tissues mapping centers have slowed down due to COVID, what can we accomplish by bringing some of these data sets together? We've talked about doing it with HCA or others anyway, but now may be a really good time to look at that and what we do by feathering existing projects and also data that may be projects that are sunsetting. So I'll leave it there.
Jason, did you need to add anything, or are we good?
Quick promotion for what Nick just said. If you want to see the call, information about that, it's in 17C. That was our breakout group. And just to emphasize our focus on practical examples of interchange as opposed to declaring standards and passing them around, hoping they'd be adopted, but to do work, in fact, inspired by the initial plenary where it was clear that different data sets were being integrated. Use that type of a very practical approach to make some progress over.
Thank you, Jason. Marianne, you're next, and Ambrose Carr is on deck. Marianne,
Okay.
are you with us? If you
I
are,
am.
go
Can you
ahead.
hear me? All right. I was moderating a session called Fair Assessment or Metrics with the subtitle of Defining a Fair Baseline. This was part of the fairness group. We started that in plenary by going in depth into the 15 FAIR principles, not just the high level FAIR, but all of the what those mean. There was another group that delved very heavily into some of the standards and other ways of interacting over FAIR, but our job was to consider whether we wanted to have any sort of formal assessment or metrics. And as we believe that assessment goes hand in hand with education, we actually focus much more on education rather than how we administer tests. So it was generally agreed that because everybody agrees that the FAIR principles are aspirational, and so one wants to take a layered approach and define simple ways to get going on FAIR and then layer on additional complexity as we go. We very much like the idea that there should be some sort of persistent structure across these consortiums that dealt with education and adoption and promotion of FAIR. So somebody brought up the customer success committees that they have at the Broad Institute, but something, again, cross consortia. We thought that a good entree into FAIR was to focus on data citation, so formally citing data sets, because that gets into how repositories deal with persistent identifiers, machine readable metadata, basic metadata, and standards. And so we want to have an activity based on data citation, much as we did in the BRAIN initiative, bring in journals, bring in researchers, bring in the repositories. Our ask then are those who are interested in that, please join us. Also, we know that other areas of the workshop dealt with FAIR, and we'd really like you to contact us if you are dealing with aspects of FAIR so we can bring this together. And we're hoping at some point that the workshop organizers deal with how this sort of synthesis across all the groups that we're dealing with related things might act. Oh, there, I just finished. That was my timer. Okay.
Thank you very much, Marianne. Ambrose Carr is next, and Peter Sorger and Niels Gellenborg are on deck. Ambrose, are you with us?
I am, thank you. I participated in a discussion on tools to annotate cells in an automated fashion, and we focused on identifying the problems that make the transfer of annotations from labeled data to novel data sets difficult. We identified a broad set of issues, which anyone interested can read in the meeting notes, but we focused on one problem that makes automated annotation hard, and that's that while training data exists, there's a lack of a standard nomenclature for labels, which means that similar cells are often annotated differently, and this makes the data difficult to reuse and consume. So in practice, it means that annotation currently proceeds with significant manual components. Data are generated, then the computational researcher will go and comb the literature, including any previously annotated data, but that often is blocked at a relatively low level of resolution before they need to go and contact some experts who understand the cell types more deeply. So given that, our group believed that there's an opportunity to generate a more effective training set. It proposes a call to action relevant to that. So we've seen as recently as the plenary yesterday, an effective approach to harmonize annotations in the lung network, where collaboration was urgently spurred by COVID-19. Authors of that were in our session and offered to release a sort of straw person protocol that described how the iterations occurred between the computational and experts. But more broadly, we think there's an opportunity to seed discussions in other consortia or link to ongoing discussions of annotations in other groups. And so what we would like to see happen is to obtain more engagement from broad networks where we learn both who is actively working on this problem and two, who has identified or produced and curated their own training data. We think that if we can identify the relevant individuals and relevant data sets, that would be a good basis for constructing and then harmonizing some kind of effective training data set to spur computational innovation in this space.
Thank you, Ambrose. Peter and Niels are next, and Michael Haralick is on deck. Peter and Niels, are you with us?
I should be. Is that you, Niels, or?
Go ahead, Peter.
Yes, so I just want to say that Niels and I led the discussions for 17A and B, which had to do with, which was also related to what Jason just mentioned, had to do with integrated search and visualization. And I think it mimics many of the things that we've heard from Christine, Ambrose, and others, and that is in particular the need to have more effective cross-project integration, both from a technical and a data standpoint. And in particular, we had 3 points that we thought should be raised with respect to that. One is, I think, an immediate opportunity to do some cross-platform education, particularly in areas that are less well understood. The particular session I read was on search. We imagine that search is trivial, but in fact, it's a fairly complicated topic that I think is poorly understood and is going to directly impact things like FAIR data visualization. We think there's actually going to need to be a resource for curators. It could be a modest number, but individuals who actually are tasked specifically with linking the various projects together, rather than the way the individual projects have to deliver really all the way from the biological use case to the data. And then finally, we think there's a role for standing up some expert networks in particular areas of computation. Because although there's a fair amount of computational work, overall it's substantially under-resourced relative to the challenge at hand. And Niels had similar thoughts on the visualization side.
Yeah, I think we only have a couple of seconds left, but thank you, Peter. I think you made a really good point. I think from on the same side, like visualization is not as trivial as it seems. I think we need to have clear use cases, but we also need to agree on common APIs to access data and common data models to have the ability to link together visualizations.
Thank you. Thank you very much, gentlemen. Michael is next, and Neil, I believe, is presenting our final soapbox after that. Michael, are you with us?
Yes, can you hear me?
Yes.
Yeah, okay, so this was an outreach group that was sort of co-organized by Gary Bader and Christine Schwenk and myself, and we surveyed, and we had some pretty good, really nice participation from several people, including Zarina Gauss from NIH and Lisa Record and Robin Scheibeck and others from HubMap and Yoshi Endo from Recon. And basically, we sort of, the topic is very broad, but we considered the sort of topics, we didn't sub-topic a sub-session, but we addressed all these topics in a single session. That one was how outreach and engagement activities can be part of single-cell mapping. The second topic is what types of technology and virtual online spaces, perhaps even lessons that we've learned in this kind of amazing exercise of the last 2 days, can sort of help to benefit this. Equity, how do we know that we're reaching all interested people and countries and continents? How are these activities to be funded? And what is our sort of roadmap for priorities? There are a lot of issues that we sort of enumerated in groups, different things, from anything from teacher curriculum to citizen science to all sorts of things, which are far too numerous to kind of go through all of them here. But one of the things I think that we would like to put forward in the spirit of the soapbox is that, does it make sense to have a kind of report? In some sense, there's a kind of an unprecedented merger of these various consortia here, and perhaps it's worth some form of announcement or write-up describing in general what was accomplished and various links to the various participants and a way to move forward. So we'd like to bring that forward as a possible action item to be voted on.
Lovely, thank you. And I believe we have Neil, who's our ultimate soapbox presenter. Neil, are you with us?
I am. Can you hear me?
Yes.
Okay, great. 2 minutes, go, soapbox. Yeah, so this was, I'm the caboose on the train. That's usually my position, so happy to bring us home. We had great energy around antibody validation to try to lift the operational baseline of many imaging technologies that utilize antibodies or affinity reagents. And so we have this spider diagram. We had this spider diagram moment where we looked at all of the pre-analytical variables and tried to categorize them that plague this general area. And so that seemed to crystallize some energy, and the second part of the breakout, we were grooving hard on that and ended up with a framework, a 5 pillars, which would be then sections in a paper that then got invited by Nature Methods associate editor, who was on and was capturing that energy and even pulling us as a community to try to wrangle with this gnarled set of issues that I won't descend into because you can't on a soapbox, but I think we have a framework together with Stephen Hewitt and Quang Hoon. I think we're gonna do it. So by 2021, you will see some type of light through the fog of antibodies, how to validate them, informed by the 3 or 4 other groups who we stand on their shoulders. So we're going to take some small bites out of a huge problem that hopefully will help 2D, 3D imaging in the future.
Nice, thank you very much. And Soapboxers, thank you for being willing to do these 2-minute presentations on the spot. It's really nice, I think, for people to get some closure and hear what was happening in some of the groups that they weren't able to go into.
Thank you, Donalyn. This is Sarah Teichmann speaking. Can everybody hear me? Yeah. I just want to say thank you to everybody for a great meeting. Donalyn's thanked the moderators, the coordinators of the breakout sessions, the soapboxers, everyone, all the participants. It has been amazing. And you can see the Twitter sphere has been alight with nice comments about this meeting. And I think the answer is that the switching to the virtual has been a success. Next slide, please. It was an experiment. We weren't sure whether it was going to work, but I think the Crocs Up has come up with some great new connections. We have succeeded in building those broader coordinations and collaborations across this amazing mapping and atlasing community, and we've come up with next steps with pilot projects. The meeting has also been a stepping stone to celebrate the advances that have been made by the research community and the different networks and consortia. And we've celebrated each other in our ongoing work, and that's been really nice and gratifying. And this virtual meeting of course, it hasn't been quite the same as meeting all you lovely people in face, but it's been pretty close. And you know, going forwards, we will try to find alternative venues to meet in person. Next slide, please. On that note, the next Human Cell Atlas meeting was going to be the really exciting development and pediatric cell atlas meeting in July in Toronto, organized by Gary and the whole committee. Now, obviously, that's going to have to shift and change in some way in time and place and so on. And we'll keep you posted. If you're interested in joining the Human Cell Atlas development and pediatric cell atlas meeting, in whatever way, shape, or form it takes place, please email meetings@humancellatlas.org. Thank you. I'll hand over to Aviv.
So we wanted to highlight a few things that really came through and many of them we've just seen in the Soapbox presentation. So there were a few common asks that we heard again and again through the different breakouts. Everyone needs an ontology. We need ontologies for cells and for tissues and for organs. They give us a joint vocabulary with which to talk to each other and also to harmonize our data set. Everyone wants to share protocols, and this includes protocols for tissue processing, for antibody validation, lab-specific protocols that could be of high interest. So one lab can be highly specialized in protocols, and others would need to learn from them. It is hard to know still where all the available data sets are and how to access them, especially as these data sets still emerge inside labs. We've seen that very live in the COVID-19 effort to pull a lot of data sets that are unpublished in order to do a meta-analysis. And it would be amazing if we had one common data type that was shared and everyone was guaranteed to collect. And one example was to collect H&E stain slides across all the consortia, sort of as a lingua franca across many different types of efforts and areas of focus. And at the same time, to be able to integrate or complement different types of data computationally, which is a big challenge for our algorithm experts. Now, there were many, many other areas that were identified. People talked about how we would handle images and share them, how we would need gene programs in order to interpret things, and there were many, many other areas as well. Next slide, please. We identified a few next steps that we can take. We're going to share as soon as possible all the information from this meeting. That includes the slides and the blue boxes and the summary, as well as the Zoom videos. I think we're at the mercy of Zoom being overloaded by everyone zooming around, so it takes a little bit of time. But all of this will appear on the website. The champions are available for you to follow, up with the group, so next steps and the next type of virtual meeting. And Meetings can be done by WebEx or Zoom, but they can also be a Google Doc or they can be an HCA Slack channel. As earlier today, Gary highlighted one for cellular ecosystems in tissues. And let us know in our email address, NIH-HCA2020@hapmapconsortium.org, of what you plan and if you need assistance. Sometimes only a day or two after the meeting you realize that you would really want to do something, so just let us know. And for the participants, if you'd like to be involved in any idea that you were part of during the meeting, contact the champion for the next steps. If you feel that there's areas in which we're missing representation, either specific experts or consortia that weren't as represented today and yesterday, or you need input from other people, just again, let us know in the same email address. And if there were any topics that we didn't discuss that you feel that you would like to champion, let us know as well. There's many other virtual ways in which we can continue. Next slide, please. And then 3 things that we hope the community would achieve soon, and by soon we mean within 6 months. Maybe have a common list of validated antibodies and methods for validation of reagents, having standard QC metrics for single-cell and single-nucleus RNA-seq. This is pretty advanced, and we hope to have the same for single-cell attack-seq, and maybe a roadmap for other types of data. And then share our current practices. Maybe they're the best ones, maybe new and better ones will be invented in the future for metadata QC, for constructing containers, for cell segmentation, and for API designs. And with this, I'm going to turn over, I believe, to either Richard or Shannon for the next slide.
Thanks, and thanks everybody for joining this meeting. As mentioned earlier, it was experimental. We weren't quite sure how it was going to work out, but we would like to thank everybody who's made this possible. So everybody from the COVID-19 presenters at the start of the meeting, for bringing together a presentation on very short notice and highlighting the work that they've done, but also framing this meeting and what can be done when members of the community get together and work together. The New Innovation Facilitation Team for making this meeting possible and helping steer the virtual space to where they need to be, to all the breakout moderators as well as the scribes for capturing the discussions that have happened over the past 2 days. And to all of you for attending this meeting. You know, as we mentioned at the start of the meeting, this is one of many meetings that are happening. And it's one of a series of stepping stones to help bring the community together. And as part of that, we had the series of pre-meeting webinars in advance of this meeting, and we'll consider different options for follow-up. I think Kadi Borner gave a great example of having a debrief for the group that met during this meeting, and you know, we would certainly encourage other groups that got together and identified both a common need as well as champions to get together themselves following this meeting. But we're more than happy, and we intend to put resources into making some of these collaborations happen. So I'd also like to thank my fellow members of the program committee who represent many different areas of our different consortia. So Ed, Aviv, Sarah, Shannon, and Young, and as well as the planning committee who helped really do the logistics and get everything together, particularly over the last month as we moved from an in-person meeting to a virtual meeting. So next slide, and final slide. So thanks everybody for participating over the past 2 days. We do want to see the work that's been discussed over the past 2 days continue. So please let's keep the discussions going. Make sure if you do hit any roadblocks to reach out to us, because we do want to see some of these ideas moving forward. So thanks again for taking the time to join us over the past 2 days. It's been a great meeting and I've certainly learned a lot. So thanks everybody for your time.